basic mechanisms of genetic pollution is simple. When a plant or animal that carries engineered or selectively bred genes produces with a wild relative, its genetic material may be passed on to offspring. This gene flow can happen through wild insects, water. No, this flow can happen through wind, insects, water, or animal carriers. In the case of plants, pollen from a genetically modified crop can travel and fertilize wild or native plant varieties, introducing genes that were never part of the wild population's evolutionary history. Similarly, domesticated animals released into the wild, intentionally or accidentally, can breed with their wild counterparts and introduce altered traits into the population. One of the most prominent and controversial examples of genetic pollution comes from genetically engineered crops. In many countries, especially those with large-scale industrial agriculture, crops like corn, soybean, canola, and cotton have been genetically modified to resist pests, tolerate herbicides, or even improve yields. These modified plants can and often do cross-pollinate with nearby non-modified or wild relatives. Once the modified genes enter the wild gene pool, they are difficult or impossible to remove. This has led to legal, ecological, and agricultural challenges. Organic farms, for instance, face the threat of contamination, which can disqualify their produce from organic certification and affect marketability. The ecological consequences of genetic pollution can be profound. One major concern is the potential loss of genetic diversity. Wild populations rely on a broad genetic base to adapt to changing environmental conditions, resist diseases, maintain overall ecosystem balance. When foreign genes, especially those tailored for specific traits, enter a wild population, they may outcompete or replace native genes. This can lead to what is known as genetic swamping, where the wild genome is gradually diluted or replaced. Over time, this reduces the evolutionary flexibility and resilience of the population, making it more vulnerable to disease, climate change, and other pressures. Another risk involves the creation of hybrid organisms that possess a mix of wild and domesticated traits. These hybrids may have unpredictable fitness in natural environments. In some cases, they may be less capable of surviving and reproducing, leading to a decline in the overall population. In other cases, they may be more aggressive or better adapted in ways that allow them to dominate local ecosystems, outcompeting other species and upsetting the ecological balances. For example, a genetically modified fish that grows faster than its wild relatives could outcompete them for food and breeding space, leading to population crashes. Genetic pollution also poses risks to indigenous knowledge systems and cultural heritage. In many parts of the world, traditional farming communities have developed and maintained unique crop varieties over generations, adapted to specific soils, climates, and cultural needs. These heirloom varieties represent a deep reservoir of genetic diversity and cultural identity. When genetically modified crops are introduced nearby, the risk of cross-pollination threatens the purity and uniqueness of these traditional strains. Once contamination occurs, it may not be possible to restore the original variety, resulting in permanent loss of agricultural heritage. Legal and ethical issues further complicate the picture. In some cases, companies that hold patents or genetically modified organisms that hold patents on in some cases companies that hold patents on genetically modified organisms have pursued legal action against farmers whose fields were found to contain patent genes. Even if the contamination was accidental. This has raised serious questions about responsibility, ownership, and liability. Should a farmer be punished for genetic drift they did not intend or control? Should corporations bear responsibility for preventing their proprietary genes from spreading into ecosystems and private lands? Efforts to prevent genetic pollution are ongoing but face many challenges. Isolation, distances between genetically modified and non-modified crops are one strategy, but pollen can travel long distances depending on wind patterns and pollinators. Physical barriers and buffer zones can help, but they are not foolproof. Seed banks and preservation programs aim to conserve pure genetic lines, but these too require rigorous protection and documentation. In animal populations, preventing escape from fish farms or livestock enclosures is critical, but escapes still occur due to natural disasters, human error, or infrastructure failure. Some scientists have proposed genetic use restriction technologies, also known as terminator genes, which are designed to prevent modified organisms from reproducing. These methods, however, have faced ethical opposition and have not been widely adopted. Others suggest that better labeling, stricter regulations, and more transparent risk assessment are needed before genetically modified organisms are introduced into new environments. It is also important to distinguish between intentional genetic modification and more traditional selective breeding. Though both can result in gene flow, selective breeding involves choosing parent organisms with desired traits to produce offspring with those traits. Over time, this can create new breeds or varieties that differ significantly from their wild ancestors. If these domesticated forms escape or are released into the wild, they too can affect native gene pools. In many cases, the results are not beneficial. Wild trout populations, for example, have been weakened by interbreeding with domesticated trout used in fish stocking programs, leading to reduced survival and reproductive success. Beyond agriculture and aquaculture, genetic pollution has implications for conservation biology and the protection of endangered species. Efforts to breed endangered animals in captivity sometimes results in the introduction of non-native genes, either through hybridization or poorly documented lineages. Reintroducing these individuals into the wild may compromise the genetic integrity of the remaining wild population. Similarly, planting non-local seed variations in restoration projects can lead to genetic homogenization, where local adaptations are lost. In the broader philosophical sense, genetic pollution reflects a fundamental tension between human technological progress and the natural world. Our ability to manipulate genes has far outpaced our ability to foresee and control the long-term consequences of those manipulations. As with many other environmental issues, the damage is often subtle, slow-moving, and difficult to reverse once it begins. The challenge lies in balancing innovation with caution, recognizing that once a gene escapes into the wild, it becomes part of a larger ecological story that cannot be undone.